So first, let me be very clear. It was not rushed. There was over a decade of research looking at the structure of viral proteins from other viruses, such as HIV, RSV, MERS, and SARS. We learned a wealth of information from studying these proteins, which positioned us to be able to move very quickly once SARS-CoV-2 came out.
Back in December of 2021, the FDA authorized two vaccines to fight against the effects of the COVID-19 virus. While at the same time, some variants started to circulate, and those variants might have affected the efficacy of the vaccines. But around the same time, there was a lot of skepticism as to whether or not the COVID vaccines were rushed. Moreover, the pandemic produced a lot of ever-changing whirlwind of information, and this influx of information in part raised a lot of concerns about the vaccines.
My name is Robert or Bob Fischetti. I work at Argonne National Laboratory in the Advanced Photon Source. My role here is I'm the Life Sciences Advisor to the APS Director, and I also manage some what we call structural biology beamlines. They're funded by the National Institutes of Health for looking at the structure of biological molecules. The APS, the Advanced Photon Source, it's constructed and operated at Argonne National Laboratory, and it's funded by the U.S. Department of Energy. It's a national user facility. So scientists, which we call users from around the US and the world internationally, can come and use the facility for research on a wide variety of topics. So the APS is one of the premier, what we call light sources. It provides ultra bright, high energy X-rays to look at the structure of the viral proteins. What we do is we synthesize proteins, crystallize them, and if we get a sufficiently large crystal, we can then use the intense X-ray beams that we have here at the APS and create what we call a scatter pattern or diffraction pattern. We look at how the X-rays are reflected or bounced off of the crystal. We measure the intensity and the positions of those on a detector. From that, we can back calculate what the structure of the protein is. Once we know the structure of the protein, we can then start looking at sort of a lock and key model as how does a particular drug bind to the protein and block whatever this particular function is. In the case of SARS-CoV-2, one of the things that's very important, one of the main targets is a protein called a protease. It initiates the process of the viral replication within the cell. If we can jam that lock and prevent that protein from doing its function, we can stop it from replicating and spreading within the cells.
Can you speak to a little bit about whether or not the vaccines were rushed?
Let me give a little biology intro. Our cells contain DNA, which is the genetic information that makes up all the proteins that our body needs to build and to maintain our cells. DNA is transferred into what's called messenger RNA, which is instructions for how to make a specific protein. So take a carpenter, for example. He might want to make a piece of furniture. Maybe he wants to make 100 of them. So he might go and cut out some template. And so he's taken the instructions, the DNA, converted into a template, the mRNA, and now he can go and make multiple copies of this. So that's one way to think of how a cell's genetic information is then interpreted and produces proteins. In our case, what we're interested in is mRNA vaccines. There's been a lot of research going on for years of how to take this mRNA, this template, and make a vaccine for a specific virus. One of the things that's important to keep in mind with the mRNA vaccines, when we get a shot with the vaccine, the mRNA is delivered into our arm. The mRNA then gets into our cells, providing the template or instructions to generate the spike protein. Once the protein has been produced, the cell then degrades the mRNA. It does not stay in our cells. It does not get into our own DNA. It does not become part of our life cycle, part of our chemical makeup. It's just a temporary infusion to protein so that our immune system can recognize the virus and therefore attack it and prevent us from getting further illness. One of the things that happened early in the pandemic is in early January of 2020, the genomic sequence for SARS-CoV-2, the virus that causes COVID-19, was released. Knowing that, researchers were able to immediately predict what the mRNA molecule that we want to generate, have our bodies generate in response to the vaccine, so that when we are infected, our immune system knows how to attack it. They knew that within a few weeks. And so now the next step was to be able to generate the mRNA, which they did pretty quickly, and then to start going through trials. And the trials, in a sense, were accelerated, but they still went through the same rigorous safety and efficacy trials as any other drug or vaccine would. They moved quickly because of the urgency, but also because there were so many trial candidates, unfortunately, because the virus was spreading so rapidly. So while it may seem like the vaccines were developed overnight, it was built on a decade of research that where we systematically learned new things that we needed to be able to do to create vaccines that are very effective and then to be able to roll them out quickly. All the machinery was already in place. It was perfect timing in a sense.
How does that relate to the genomic sequence of the virus?
Knowing the genomic sequence of the virus, we could immediately look at previous viruses like MERS and SARS, which are similar. And I think about 70% of the genomic sequence is the same between them. So there's some differences in proteins within the different viruses. Knowing where the differences are gives us information about why this one might be so infectious or why it may be causing disease in a different way than the other viruses did. So there's information that we can learn from the genomic information, but it also guides us in terms of trying to look at, okay, what is the structural difference? Now that you know the sequence of amino acids strung together, you can think of putting beads on a string, and the beads have different shapes. And once you build up this protein as a string of beads, it then folds into a particular conformation. But if you change one amino acid that can change, or a few, that creates a variant and that can change its structure. And so there are some differences in the previous viruses and the SARS-CoV-2, which create the significant differences. But then there's also some minor differences that occur, minor substitutions of amino acids that generate the variants that we're seeing now.
When a variant appears, is it easy to then edit the vaccine so that it can target those variants? Or how does that work? How do different vaccines target different variants?
Once you know the genomic sequence of the variants, now you know where to look in the proteins to see where did the changes occur. So for example, you can say, okay, there were several changes in the spike protein. We know we need to go and look at the structure of the spike now from this new variant and see what changes do we need to make in the antibody so that it still effectively binds to the new spike protein. And one of the things that's recently come out as the so-called bivalent mRNA vaccines. What they're doing is it's two components. It's one that retains the original mRNA to generate the antibodies against the original SARS-CoV-2. And it also has a component which will generate antibodies against the Omicron variant. So it's providing double protection. Ongoing research is what led us to this point. Had we stopped doing research on viruses like MERS and SARS because they were no longer circulating, we would not have been in the position that we were where we could rapidly develop the vaccine. It's important to continue doing basic research. I also want to stress that at Argonne and APS, there's a lot of other types of research that go on. There's research on the structure of batteries, trying to make batteries 5 times more efficient, to be able to charge more quickly, to last longer. There's a lot of research on solar cell technology and in material research areas. And one that I mentioned earlier was studying air turbines in jet engines, trying to understand how they fatigue to make a better jet engine, make it last longer. I think it's just important to know that basic research often takes a long time to yield the information that you need. And had we not been doing research on vaccines or on viruses like SARS and MERS in the past, we would have nowhere been in the position to rather quickly develop the vaccine that is needed for things like COVID. There may well be other pandemics down the road, and we need to keep doing research to be prepared for the next one.
Hi, this is Denise from C2ST. Thanks for watching. If you liked the video, please make sure to follow, like, and subscribe. Also, be sure to hit the bell icon so you'll be notified when a new video is posted. Doing all of this makes sure that our videos are more visible to other STEM enthusiasts on YouTube.